All right. Sorry for the technical difficulties we had. Had a little bit of a delay, but we're ready to get started. So thank you all for coming today and joining Melissa Naugle's thesis defense for her Master's of Science degree in Marine Science at Moss Landing Marine Labs. My name is Cheryl Logan, and I'm an associate professor in marine science at California State University, Monterey Bay, and I'm a consortium faculty member at Moss Landing Marine Labs. I've served as Melissa's primary research advisor for her thesis work that she'll be presenting to you today. And also joining us is Amanda Kahn, who served as Melissa's Moss Landing co-advisor, and Max Grand, who is her third committee member. So welcome to everyone. Before I jump into Melissa's official introduction, I'm gonna just briefly talk about how the virtual seminar is set up today. So as you probably have already noticed, all the audience participants are going to be muted throughout the entire presentation. And we ask that you please do not attempt to turn on your video or share your screen during the talk. There will be an opportunity for all of you to ask questions to Melissa at the end of her presentation. We will ask you to use the Zoom raise hand feature. This is going to be located under the reactions tab at the bottom of the Zoom window. The chat will be turned off, so if you do type anything into the chat, it won't go out to everyone. It's just going to go to the meeting host. So we do ask that you use the raise hand feature. And after the Q&A session, I'm going to ask everyone to unmute, turn on their videos so that we can give Melissa one last awesome round of applause before we do the closed door committee session, at which time we'll ask all of you to sign off of the Zoom meeting. So it is my pleasure, my great pleasure, to introduce Melissa Naugle's thesis defense. Today, Melissa is going to be discussing how prior exposure to pollution stress affects corals' ability to withstand heat stress. And her research was really motivated by some intriguing preliminary data that myself and colleagues had collected, showing that corals living in more polluted areas may be more thermally tolerant. And this research was conducted in American Samoa using a coral species called Acropora hyacinthus, which is a widespread reef building species within the Indo-Pacific. So Melissa was very excited about these preliminary and kind of contradictory findings to what we might expect, that exposure to stress might actually help a coral withstand future heat stress. And so Melissa independently wrote several grants that supported her travel back to the same field sites that we had been to previously to follow up on these findings for her master's project. In this process, she recruited 2 undergraduate scholars from our Undergraduate Research Opportunity Center at CSU Monterey Bay to join her in this adventure. And they completed their field season during a 3-week time frame in August of 2019, so prior to the pandemic. And as you will see today, Melissa has completed just an immense amount of work, including leading this remote field work, and collecting corals and running field-based assays. She then brought these coral samples back to CSU Monterey Bay and Moss Landing to investigate the mechanisms underlying temperature stress using RNA sequencing. This required some really high-level programming and bioinformatics skills that Melissa picked up very quickly, especially as a master's student. And so before I hand it over to Melissa to describe her research in more detail, I want to also describe some of the amazing accomplishments that Melissa has had while being a student at the Moss Landing and CSU, within the CSUMB and Moss Landing programs. So Melissa grew up on the East Coast in Maryland and attended college at the University of Maryland College Park. It was there that she earned a BS in environmental science and policy with a concentration in biodiversity and conservation biology. After graduating, she then did a number of research experiences where she was working on coral biology and coral conservation in Palau, Western Australia, and Thailand. And now is even a co-author on a publication that came out of some of the work she started before becoming a master's student. She then joined my lab at CSUMB in the fall of 2018, initially as part of the environmental science program at CSUMB, and then because of her interest in marine science, transferred into the marine science program at Moss Landing starting in 2019. And as I said before, she's really been interested in how local and global stressors are going to affect coral reefs into the future. And Melissa's going to be starting a fully funded PhD program in just a couple of months. So the timing of her master's is fantastic. So as a student, she's grown into a budding scholar. She has presented at 2 international scientific meetings. In fact, just this last week at the International Coral Reef Symposium, and earlier this year at the Virtual Society for Integrative and Comparative Physiology. Sorry, that's supposed to be biology, the SICB meeting. She's also been prolific in her ability to write grants and get her research funded. So I'm just listing a few of the scholarships and awards that she's had, including the John Martin Scholarship this year. She was awarded the NSF Graduate Research Fellowship Honorable Mention last year. She also was awarded the CSU Coast Graduate Award, the Women's Leadership Council Scholarship, the Myers Oceanographic and Marine Biology Trust Award, the Explorers Club Marmont Scholars Grant, as well as a Marine Interinstitutional Synergistic Grant to support some of the outreach work that she's done in the Monterey area. Melissa's also been just a fantastic teacher and mentor to our students at CSU Monterey Bay. She was a TA for our introductory biology course for 6 semesters. And in addition, she was a TA for my upper division marine experimental physiology course for 2 semesters, in which many of the students got to participate in her research by doing some of the RNA sequencing work that supported her thesis. In addition, she was a direct mentor to 4 of our undergraduate research opportunities, or UROC scholars, including Jenny and Casey, who are shown here, that Melissa brought with her to American Samoa for her field work. And Sylvia and Amalia were 2 other students that she's been working with more recently with some of the molecular lab work that she'll be presenting today. Melissa's also been a fantastic role model in the marine science community in Monterey and in California. She's the president of the Future Females Marine Science Outreach Program, which she helped design with other students as part of the Marine program. She was the Moss Landing Student Body Intercampus Liaison, in particular facilitating the relationship between CSUMB and Moss Landing. She was the co-chair for the Monterey Area Research Institutions Network for Education. And she was also selected as a science translator through the California Council on Science and Technology Science Translator Program. There's some images of her here presenting in Sacramento on her work. Melissa's also just been a really fantastic member of our lab. She's a great team player and contributor. She served as our lab safety coordinator and has been really dedicated to helping us as a lab develop open science best practices. She always is very positive and has an upbeat attitude and has provided a lot of creative and constructive input to her peers and has always been just super generous in her time helping others in the lab. I also just want to point out that during the pandemic, Melissa also volunteered her time for the past year and her molecular biology skills to help run COVID tests at the Monterey County Health Department. And this is the last slide before I hand it over to you, Melissa. We just, as a lab, want to say good luck to you in your future studies in the PhD program that you'll be starting at Southern Cross University in Australia whenever the border opens and you can actually get there. And we will dearly miss you. So with that, I'm going to hand it over to Melissa to share her research.
Thank you, Cheryl. That was a great introduction. I very much appreciate the good wishes. Okay, so thank you again to everyone for taking some time out of your day to learn a bit more about how pollution affects coral thermotolerance. So before I get started, just add this. Okay. So as we're all probably aware, the global climate is changing due to human-caused carbon emissions. And this is causing an increase in the frequency and the severity of these temperature anomalies that we're seeing more and more over time. And this is pushing many species past their physiological limits, which can cause their populations to decline or to eventually become extinct, which is leading to this major decline in biodiversity worldwide, which is a huge problem. And as species begin to approach these physiological limits, they have a few different options that they can use to persist. So I've made this cartoon featuring this lovely penguin here to show you 3 of these options that species can use when climate change begins to cause them a lot of stress. So first, if temperatures are rising too much, species can migrate to an environment that's more favorable. So for example, this penguin can just move somewhere colder. Secondly, if migration isn't an option, species may use adaptive evolution to evolve towards a genotype that better suits their environment. So, for example, if temperatures are rising, certain individuals in a population will be better suited to their environment and they'll persist due to survival of the fittest. So, future generations will be better suited to warmer temperatures. Third, so while evolution happens across generations, phenotypic plasticity can occur sometimes in just one generation. And so this is when we have non-genetic changes that allow an organism to persist in a warming environment. So you can kind of think about this in a similar way that you might acclimate to a new environment. So for example, you might take a vacation to the mountains, maybe there's high elevation there, and you might feel sick at first, but over time your body will acclimate and you'll start to feel better. So in the same way, species can use these non-genetic mechanisms to better tolerate rising temperatures or climate change over time. And phenotypic plasticity is going to be the focus of today's presentation. And I'll be using coral as a model organism to study plasticity responses to climate change because corals are stationary and they can't migrate. They also live near their thermal maximum, and they have seen these major declines recently with within species variation in survival. And that variation in responses to climate change may be due in part, at least, to these differences in plasticity. And also, in addition to just being a great model organism, corals are really important to humans and serve many functions. So we want to study them so that they can persist into the future. So corals have all these different ecosystem services that are important for humans. They support a huge amount of biodiversity. They support fish habitat that provides food for many people. They support the tourism industry. They protect our coastlines, and all of these things are really important. And we want to make sure that corals can continue serving these functions in the future. But the issue is that corals are bleaching. So this typically happens due to rising sea temperatures, but it can occur due to other stressors sometimes too. So coral bleaching is something you may have heard about before, but I'll kind of break it down for you now. So what happens in coral bleaching is there's a disruption in the coral symbiosis with its photosynthetic algal symbiont, sometimes called zooxanthellae, though I'll be calling it just a symbiont for the rest of the presentation. So when you have a healthy coral, it has typically a lot of color, and that's because it has these photosynthetic symbionts that live within the coral cells. And these symbionts provide over 90% of food for the coral. Then when the coral comes under stress, they'll expel these algal symbionts, they'll lose a lot of their color, but more importantly, they're losing their primary food source. And that's what happens when they become bleached. Then if the bleaching persists for long enough, or if the stress is severe enough, then the coral that is bleached can eventually die. And so that's what's happening here. The coral overgrows with a different type of algae and can no longer perform those ecosystem services that I showed you in the last slide. But interestingly, not all corals bleach the same. So even corals of the same species on the same reef exposed to the same environment can have different responses to heat stress. So here are some images just showing you what that might look like. So here is a healthy Montipora right next to a bleached Montipora. So they're on the same reef, but they are having different responses to their environment. And then in this other photo, we have a healthy Poecilopora next to a bleached Poecilopora next to a dead Poecilopora. So now we want to know, why is this happening? And maybe plasticity is involved in these different responses to environment. So now I'll go into two different mechanisms of plasticity that corals can use to increase their thermotolerance. So the first method that I'm going over is called symbiont shuffling or symbiont shifts. And I have a little cartoon to show you how this works. So say you have a healthy coral here, and within that healthy coral, there's an entire community of symbionts that live within the coral cell, so different species. And some symbiont species can promote higher coral thermotolerance than other symbiont species. So in this example, we have this blue, more thermotolerant species, and this pink, less thermotolerant species. Then, say, for example, this coral comes into some stress and becomes partially bleached, so it's going to expel many of its symbionts. But then say the coral recovers. So when this coral is recovering, it's now shuffled its symbiont species, or its symbiont community, sorry. And now it has just this more thermotolerant blue species. So this is how symbiont shuffling works. And this has actually been observed and measured in the real world. So this is a study from the Great Barrier Reef, where they showed that before a bleaching event, corals hosted two different symbiont species, this more heat-tolerant Durostinium and this less heat-tolerant Cladocopium. Then there was a bleaching event, and after the bleaching event, the corals hosted just this heat-tolerant Durostinium. And this shift in the symbiont community increased the coral thermotolerance by up to 1.5 degrees Celsius, which is actually quite a bit. So now on to the second mechanism of plasticity. So this is called gene expression. And to understand how this works, first you have to realize that all corals and all other organisms are expressing certain genes all the time. And this is what allows them to make the proteins that they need to function. So this baseline level of the amount of gene expression that's going on is what we're going to be talking about here. So when corals are under mild stress all the time, their baseline gene expression may involve making extra copies of gene transcripts involved in the stress response. And when they have this mild baseline stress, that can prime or can prepare gene expression pathways to allow the coral to tolerate future stress a little bit better. So here's a image showing how that might work. This is from the study here. So basically, if you have a coral that is not stressed, and then you have a coral that is under a mild amount of stress at baseline levels, they're going to be exhibiting different baseline gene expression. So the baseline gene expression of the coral that's not under stress is not going to involve making that many gene transcripts of stress-related genes. Compared to the coral that is already under mild stress, that coral is going to be making more gene transcripts of stress response genes. Then say you take both of these two corals and give them some heat stress, say they are exposed to a bleaching event. The coral that has not already been stressed prior to that bleaching event is going to have to really ramp up its stress response gene expression in order to respond to that bleaching event. And that means there's going to be a big difference between its baseline gene expression and its heat stress gene expression. And because there's such a difference there, that means that that coral is going to end up bleaching. Compared to the coral that's under mild stress and is already expressing quite a few stress response genes, it doesn't have to change its gene expression all that much under heat stress, and therefore that coral is not going to bleach. So we just learned how symbiont shuffling and how gene expression shifts can improve thermotolerance when we have a mild stressor followed by more significant stress. But when we have multiple stressors, we may find that these mechanisms work similarly or entirely differently. For example, one stressor might assist an organism in responding to the second type of stress, or one stressor might prevent the organism from responding as well to the second stressor. So today we're going to be examining these possibilities on corals using two common stressors found on coral reefs: nutrient pollution and heat stress due to anthropogenic climate change. And nutrient pollution is an interesting type of stress to study. It's been shown to have detrimental effects on coral reef ecosystems as a whole, but its effects on thermotolerance, coral thermotolerance, is a little bit less clear. So some studies have shown that nutrient pollution actually decreases thermotolerance. So here are a few recent studies showing examples of that. In contrast, some other recent studies have shown that pollution can have the exact opposite effect and can actually increase thermotolerance. So again, here are some examples of recent studies showing that. So we want to know how nutrient pollution is affecting thermotolerance, and we are choosing to study this question in American Samoa, specifically on the main island of Tutuila. So American Samoa is right here in the middle of the Pacific Ocean, and it's a great study site because it holds reefs that have been impacted by pollution, not too far away from reefs that are nearly pristine. It's also a great place to study these questions because it's an American territory. So land-based pollution on Tutuila stems from three main sources: agriculture, pig farms, and inadequate waste disposal. And so here is a figure from this paper here showing the dissolved inorganic nitrogen load at each watershed across Tutuila. So these darker areas are more impacted, areas with higher nutrient pollution, and they're not too far away from places that are not too impacted, as you can see here by the paler purple color. And we'll be studying the impact of pollution on the thermotolerance of this beautiful branching coral here called Acropora hyacinthus. This is a cosmopolitan coral that is found across the Indo-Pacific, and it can be classified as a weedy coral because it is more sensitive to bleaching than other coral species, but it also tends to grow faster. Also, it's pretty well studied at both ecological and genomic levels. And so all of these factors make Acropora hyacinthus a great candidate for this study. So now on to our research questions. The first thing we want to know is there any variation in thermotolerance of Acropora hyacinthus by pollution level? And the first possibility that we might find is that thermotolerance does not vary by pollution level. And this could be because thermotolerance does not vary at all, or because it varies by some other factor other than pollution level. Secondly, we might find that polluted sites hold more thermotolerant corals, or we might find that polluted sites hold less thermotolerant corals. So now we'll look at these three hypotheses and how they could be explained by symbiont community. So if we find that thermotolerance does not vary by pollution level, it might be because we see no symbiont community differences by pollution level. If polluted sites hold more thermotolerant corals, it might be because pollution is causing shifts to a more heat-tolerant symbiont species. Third, if we find that polluted sites hold less thermotolerant corals, it might be because pollution is causing shifts to a symbiont species that is less heat tolerant. So now again, let's look at these three hypotheses and how gene expression patterns may explain them. So if we find that thermotolerance does not vary by pollution level, it might be because there are no differences in baseline gene expression before heat stress. If we find that polluted sites hold more thermotolerant corals, it might be because pollution primes gene expression to heat stress because the baseline stress level due to pollution stress is causing an increase in stress response gene expression, which prepares the coral for subsequent heat stress. Third, if we find that polluted sites hold less thermotolerant corals, it might be because there are different genes needed to respond to pollution stress versus heat stress. So the baseline stress levels may actually be working in the opposite direction. And so while we may expect to see any of these three hypotheses, there's one in particular that we may favor, and that is because of this preliminary study that Cheryl mentioned in her introduction, where in 2014, they studied on Tutuila in a Cropper hyacinthus, the same species in the same site, how pollution affects thermotolerance. And they found that at high pollution levels, the bleaching threshold was much higher, meaning the corals were more thermotolerant, which is a bit unexpected. And they found that the more polluted sites hosted more thermotolerant corals because they held more thermotolerant symbiont species. So those symbiont shifts were really accounting for the differences that they were seeing in thermotolerance. Yet they also found this unexplained benefit of pollution on thermotolerance that could not be explained by the symbiont shifts. And so now we want to know maybe gene expression responses to pollution could help explain this unexplained benefit of pollution. So I examined these questions again on Tutuila, American Samoa, and these are the 5 sampling sites that I chose. We have 2 high pollution, 1 moderate pollution, and 1 low pollution site. And these pollution categories were determined using this dissolved inorganic nitrogen load, but also using data from American Samoa EPA pollution designation, protection status, and human population level. So now I'll go into each of these sites a little bit more just to orient you. So first down here, we have Fongatelle on the south side of the island. This site is relatively pristine. It's set in the NOAA National Marine Sanctuary, and that means that it has a certain protection status. Up on the north side of the island is Vatia, and this is set in the National Park of American Samoa. It also has a relatively low population level, though not as low as Fongatelle. For our high pollution sites, the first one we have here is called Cannery, and this is the site of the star-kissed tuna cannery. So if you've ever eaten this tuna before, it was canned right here. And just to really drill it in how polluted this area was compared to the low pollution sites, we sampled coral that were like right behind this building here. And then our second high pollution site is called Coconut Point, and this is the site of the Pongo Pongo International Airport. And so the corals we sampled were right off of this airline landing strip. Okay, so at each site, we sampled our Cropper hyacinthus on snorkel so that we could assess thermotolerance differences using our heat stress assay. And this is the heat stress assay. It's called CBAS for short. It's the Coral Bleaching Autonomous Stress System. And this is just a time-lapse video showing all the work that really went into getting the setup, debugging it, putting it all together. And what this is 8 different tanks, each set up with a light, a heater, thermometers, and a cooling system. All of this is controlled by an Arduino computer. And this is what it looks like in action. So this is in American Samoa, and you can see in each of these tanks a bunch of coral nubbins being exposed to heat stress. And this assay is great because it is cheap, it's portable, it's ideal for experiments in places that do not have marine stations. So here's a breakdown of the experimental design of those 8 tanks. So first we have a tank at 28 degrees C, and this is our control tank because it's at the same temperature that the corals would be experiencing on the reef at that time. And then we have 3 experimental tanks at 33, 34, and 35 degrees C. The entire thing is replicated twice. And then within each tank, we have 16 different corals from 8 different colonies. And then in each of those experimental tanks, there's a ramp hold heat stress assay that's performed. So this is a figure showing that ramp hold. The way it works is each morning we would sample the corals, bring them back to the site of our heat stress assay, and we would place them in the assay right around noon. We would allow them to acclimate to the control temperature at 28 C for an hour, and then they would spend 2 hours ramping up to temp. Then they'll spend 3 hours at their temperature and then ramp down over the course of an hour. Then they'll sit for an overnight recovery at 28 degrees C. So this makes up a coral stress test that can assess thermotolerance differences. And it's been shown in the past to be able to discern differences in thermotolerance among reefs, just as well as a more traditional longer-term heat stress assay. So now that at this point I've gone through the introduction and the methods for the heat stress assay, now I'll go through all of the methods, results, and interpretation for our measurements of thermotolerance. So measuring thermotolerance, we did this using 3 different methods. The first 2 measured color paling, or basically how bleached the corals were. And the third measured the performance of the symbiont. So I'll go through each of these a little bit more now. So our first method of color paling is this Coral Watch Health Score. So this is a card that you can place right next to the corals underwater and basically perform a color match to determine a score that represents how bleached the coral is. So this is a quick and easy metric to determine visible bleaching. Secondly, we used red intensity. So this also measures visible bleaching, but in a bit more objective way. So what we do here is we take a photo of the coral nubbins next to a grayscale, and then we put all of these images into a computer program that normalizes them based on the grayscale and then calculates the intensity of the red within each coral nubbin. So basically, if the red intensity is increasing, that means the corals are more bleached. And then third, we measured the symbiont performance using photochemical efficiency. So this uses something called pulse amplitude modulated fluorometry, or PAM for short. And it basically is measuring how efficiently the symbionts are photosynthesizing, with the idea that as the coral and the symbionts become stressed, they're not able to photosynthesize as well. So this can detect photochemical damage even before visible bleaching occurs. So now that we've gone through these 3 different methods of thermotolerance measurements, I'll show you the results. And I have them here all summarized on one convenient table. And what you might notice is there's a bit of variation and a lot to take in, so I'll break it up one metric at a time. So first we have Coral Watch Color Card Health Score. And this is at our 2 high pollution sites, our moderate pollution site, and our 2 low pollution sites. And in each box, you'll see either a dash, meaning that site had corals that were not significantly different than other sites, or you'll see a pale coral indicating that corals at that site bleached more, or you'll see a healthier, darker coral, meaning that corals at that site bleached less. So when we're looking at the color card health score here, our first metric, we see that at Vitia, a low pollution site, the corals bleached less than these 2 other sites at moderate and high pollution levels. When we look at our measurement of red intensity, we again see that Vitia, a low pollution site, bleached less, this time bleaching less than another low pollution site and a high pollution site. Finally, when we look at Symbiont performance, this time we see that Funga alu, a moderate pollution site, bleached less than Fungatelle, a low pollution site. And so you might be thinking there's quite a bit of variation, not sure what to take away from this. But there is one trend that sticks out, and that is right here. So Vatia, this low pollution site, and 2 out of 3 metrics tended to bleach less, while Fongatelle, again, another low pollution site, tended to bleach more in 2 out of 3 metrics. So now let's look at this by pollution level in a different way. So if we combine these thermotolerance data into a single metric, we can look for trends by pollution level. So this is our metric of thermotolerance, with points closer to here as more thermotolerant, points closer to here as less thermotolerant. And we've broken up the coral colonies by pollution level. What you'll notice is quite a bit of variation and no clear trend with pollution level. But when we compare these data back to 2014, we can see there's a bit of a difference. In 2014, again, like I said in the introduction, high pollution sites had more thermotolerant corals, but that's not what we're seeing in 2019. So just to show this in a more visual way, in 2014, polluted sites that looked more like this held more thermotolerant corals, and less polluted sites that looked like this held less thermotolerant corals. But by 2019, all sites held relatively equally thermotolerant corals, or at least thermotolerance did not vary by pollution level. And in 2014, if you remember, thermotolerance was best explained by differences in symbiont community, which naturally leads us into our next question, which is, how does symbiont community affect thermotolerance under pollution stress? So now I'll go through the methods, the results, and the interpretation for our measurements of symbiont communities. To quantify symbiont communities, I extracted DNA from the coral in the symbiont, and then used a method called qPCR to identify the relative levels of 2 of the most common species, of symbiont in Acropora hyacinthus in American Samoa. So these 2 species are heat-tolerant Durstinium and heat-sensitive Cladocopium. And now I'll show you the results. So this is a violin plot showing the ratio of Cladocopium to Durstinium, and each point represents 1 coral colony. So points closer to up here hosted fewer heat-tolerant Durstinium, while points closer to down here hosted more heat-tolerant Durstinium, and points at this -20 mark hosted 100% heat-tolerant Durstinium. And you can see how this differs among our sites. We have our low pollution sites in blue, our moderate pollution site in green, and our high pollution sites here. And you'll notice that our low pollution and moderate pollution sites host a combination of Cladocopium and Durstinium, while our high pollution sites are hosting 100% Durstinium. And again, to show this in a more visual way, we found that more heat-tolerant symbionts were found in polluted areas, with our polluted sites holding 100% heat-tolerant Durstinium, just this one that I've been showing as blue, and our less thermotolerant symbionts were found at less polluted sites. So the less polluted sites had a combination of heat-sensitive Cladocopium and heat-tolerant Durstinium. And this is interesting because it aligns with past studies that have found that higher levels of Durstinium tend to be favored in more stressful environments, such as areas where there is a lot of temperature variation. But it's important to consider that there are trade-offs between hosting Durstinium. So while it will make the coral more thermotolerant to host more heat-tolerant Durstinium, it also tends to make the coral slower growing. So Cladocopium may actually be more useful in certain circumstances because it tends to promote faster growth, as well as other trade-offs. And also interestingly, when we correlated symbiont community to thermotolerance, we found no correlation, which is really surprising because typically when corals host more Durstinium, that means that those corals are more thermotolerant. When we plot these symbiont proportions of Durastinium and Cladocopium together, the lack of correlation starts to make a little bit more sense. So even though we have differences in symbiont communities among sites, like you saw in the violin plot, those differences are actually quite minimal. If you really squint, you can kind of see a little pink sliver right here in these two sites. It's more apparent in Fongatellae, though that is driven by just one coral individual. So again, even though symbiont community varied by pollution level, all sites still hosted almost entirely Durastinium, again, with the exception of Fongatellae. So now let's compare our data to data from 2014. So again, this is the same violin plot that I showed you a few slides back, showing our trend of symbiont community with pollution level. And now let's compare to 2014. So we can kind of see a similar trend with pollution level, but like in both of the 2 years, where less polluted sites tend to host more Cladocopium and more polluted sites tend to host more Durastinium. But what we'll also see here is a bit of a shift in time between 2014 and 2019. In 2014, the site Vitia held many corals that hosted quite a bit of Cladocopium, though by 2019, that really dropped off. I'll also point out Coconut Point, which had a combination of Cladocopium and Durastinium in 2014, but by 2019 hosted 100% Durastinium. So what this indicates is that we may have captured a shift to Durastinium over time between the years of 2014 and 2019. So again, to show this in a more visual way, and this is what we think happened. So in 2014, we had this symbiont community of 2 different species, Durastinium and Cladocopium, in many of our coral colonies. Then we think that many of the corals became stressed and expelled some of their symbionts. Then by 2019, the symbiont community has been shuffled and is now dominated by Durastinium. And we think that this stress that may have happened, inducing this shift in symbionts, could be due to the major bleaching events that happened in 2015 and 2017, or possibly due to pollution stress as well. So while differences in thermotolerance were determined by symbiont community in 2014, there has been a shift in symbiont community by 2019, evening out most of that variation. So by 2019, thermotolerance no longer varies at the site level, though there is still variation in thermotolerance within sites. We know that variation in thermotolerance in 2014 was primarily explained by symbiont community differences, but in 2019, symbiont communities did not explain differences in thermotolerance. So we want to know maybe gene expression patterns can explain that variation. So that's what we're going to find out next. Again, in 2014, these differences in how corals are bleaching was attributed mostly to symbiont community. But by 2019, we know it's not really by symbiont community. So now we want to know, is it gene expression that can explain these differences? So that brings us to our measurements of gene expression. So now I'll go through the methods, results, and interpretation of our measurements of gene expression. And just a quick reminder on why we're studying gene expression for those of you that don't study it. All organisms contain DNA, but that doesn't mean that all of their genes are being used all the time. By measuring gene expression, we can figure out which genes are being used and how much, especially in response to their environment. So we measured gene expression in a subset of corals from control and high heat stress treatments that were sacrificed during peak heat stress. To measure gene expression, we started by extracting RNA, the same thing in your favorite COVID vaccine, and we prepared cDNA libraries and performed RNA sequencing to look at what genes were being expressed under control and heat stress conditions. Then we performed differential gene expression analysis to compare those groups and see which genes are expressed at different levels between control and heat stress, between high and low pollution, and between the most and the least thermotolerant corals. We also used a method called WGCNA, which I'll show you more later, to identify how entire groups of genes are expressed in relation to treatment, pollution level, and thermotolerance. And then finally, we did something called a gene enrichment analysis. Again, I'll show you more about that later, to identify what genes are most important and what those genes are actually doing in the coral. And so now on to our results. So first, what I'm showing you here is how gene expression differed at control versus under heat stress. And we found that it differed quite starkly. So this is a PCA plot that shows gene expression differences in 15,109 genes in something called Euclidean space, meaning that points or triangles that are closer together are more similar in their gene expression patterns, while points that are further away are more different. And what you'll notice is that on the left here, we've got a bunch of triangles representing how corals at control are relatively similar to one another. And then on the right here, we have a bunch of circles showing how corals under heat stress are grouping separately from the corals at control. So now we'll look at the same pattern in a slightly different way. This is a heat map that can be used to visualize how gene expression relates to treatment. So I'll just kind of break down this figure for you since we'll be looking at another one later. So on the left here, we have 25 different modules. And what that means is we have 25 different groups of highly correlated genes. So within each, within each group, we have genes that behave similarly to one another. Then what we did in this analysis is we correlated those 25 groups of genes to either the control treatment or the heat stress treatment. And then within each of these cells, you'll see either a dash or a number. So if you see a dash, that means the correlation between that module and that treatment was not significant. But if you see a number, that means that it was significant, and the number shown is the Pearson's R correlation value. So closer to 1 or -1 shows a stronger relationship. And then finally, you'll notice that each cell has a color attributed to it. So if that color is closer to red, that means that that group of genes was upregulated or turned up in that treatment. Excuse me. And then if you see that cell is blue, that means that that module or that group of genes was downregulated or it wasn't used as much in that treatment. And what you'll notice here is there's quite a stark difference between the colors and the numbers at control versus the colors and the numbers at heat stress. And what that means is that gene expression differed under heat stress, the same thing we saw in our PCA plot. So now I'll just kind of choose a couple of these modules or groups of genes. We have quite a few that are significant, but we'll dive into the ones that were showed the strongest relationship. So the ones over 0.9. So the first one is here, the A7 module. And this module was down-regulated under heat stress, meaning used less by corals under heat stress. And these genes were involved primarily in metabolism and development. And then now we'll look at this A15 module, and this module was upregulated under heat stress, meaning that corals under heat stress use these genes more. And these genes were involved in the immune response, in DNA maintenance, and in the stress response. So taken together, what we see is that stress response genes are observed under heat stress, which we might expect. And these patterns highlight the characteristic gene expression response to stress. Corals and pretty much all other organisms follow these patterns when they're under stress, especially heat stress. So what this typically involves is an upregulation of genes involved in mitigating and repairing cellular damage. But that comes at an energetic cost to the organism. So typically that also means that the organism needs to downregulate genes involved in growth and development in order to conserve energy. So this is interesting, and it's cool that we can measure these changes happening under heat stress, but these results aren't exactly groundbreaking. These patterns have already been well studied, but what we're really interested in this study is how baseline gene expression affects thermotolerance. So that's why we're going to look at baseline gene expression, meaning gene expression just at control conditions, not at heat stress. So we're going to zoom into that control gene expression, and we're going to ask first how pollution itself affects gene expression, and second, how gene expression before heat stress might impact thermotolerance. So first, we're looking at how baseline gene expression, meaning gene expression at control conditions, varied by pollution level. So again, remember we have these groups of genes here called modules. There's 17 of them, and we're correlating them to our different pollution levels. And what you might notice is there's one module here that has different trends in the low pollution versus the high pollution. And this module, or this group of genes, was primarily made up of genes involved in methionine metabolic process. And methionine is an essential amino acid, but what does that mean? That corals in high pollution are using these genes more. Well, high pollution corals are maybe increasing methionine production. That's what our results are indicating. And methionine has been shown in past studies to act as an antioxidant under stress and can potentially scavenge reactive oxygen species, which build up under stress. So what this indicates is that possibly pollution might be affecting corals at the molecular level, even though thermotolerance is not varying by pollution level. Okay, so now we'll look at baseline gene expression. And instead of relating it to pollution like we just did, now we're going to be relating it to thermotolerance that we measured during our heat stress assay. So we ranked thermotolerance and we identified the most thermotolerant corals and the least thermotolerant corals. And then we looked at how their baseline gene expression correlated to what group they fell into. And what you'll notice is that there's a bit of a difference between the baseline gene expression at the most thermotolerant corals versus the baseline gene expression at the least thermotolerant corals. So now we'll dive into the 4 modules here that were upregulated in the most thermotolerant corals. And these modules contained genes that were involved in cytokine production and the immune response. So that means that corals with higher thermotolerance tended to upregulate these genes even before or while they were not even in the heat stress treatment. Now let's look at the modules that related most to the least thermotolerant corals. And these modules contained genes involved in cell death and protein degradation. So what this means is that the most thermotolerant corals benefited, perhaps, from higher baseline levels of immune response and cytokine production genes even before heat stress. So that might be because these corals have been mildly stressed or something other than pollution has triggered higher expression of these genes, which may promote thermotolerance. Likewise, we also found that higher baseline expression of cell death and protein degradation genes might be detrimental under heat stress and inhibit thermotolerance. So after seeing these results, our next question would be, why are some corals expressing higher levels of these genes versus why are some corals expressing higher levels of these genes? And there's a variety of environmental, ecological, or evolutionary factors that might explain these differences in baseline gene expression. But from our study, we're seeing that it doesn't really appear like pollution has a very large effect. So now I'll bring everything together and talk about all of the results in the conclusions. So just to remind you what we found in the study, first, we found that pollution did not correlate with thermotolerance in 2019 when the study was done, even though in 2014 it did correlate. We did find that pollution correlated with a higher proportion of heat-tolerant durastinium, with polluted sites hosting 100% durastinium and lower pollution sites hosting a combination of the 2 different symbiont species. We also documented this possible shift in symbiont community between 2014 and 2019. And when we looked at baseline gene expression, we found that higher expression of certain genes, those involved in cytokine production and immune response, correlated with higher thermotolerance to heat stress, while baseline gene expression of cell death and protein degradation correlated with lower thermotolerance. What does this all mean? Why do we care? This is really important because by 2019, we're seeing that variation in thermotolerance is not really determined by a symbiont community anymore. And this may be because symbiont-driven thermotolerance is close to reaching an upper limit, since all corals hosted almost entirely Durastinium. So what that means is that the remaining variation in thermotolerance that we're still seeing today may be due in part to baseline gene expression levels, which will be especially important to consider in the future as symbiont-driven thermotolerance continues to reach this upper limit. And finally, baseline gene expression will allow management programs to identify thermotolerant corals and to target for conservation or to use in breeding programs. And with that, I would like to say thank you to everyone that has made this project possible. I've listed quite a few of you here, but apologies if I have forgotten anyone. I'd like to thank all the people who advised and mentored me to help guide this project. I would like to thank everyone who assisted in the field and everyone that helped coordinate the field research and answered all of my many questions before arriving in Tutuila. I'd like to thank everyone who assisted with the lab components of this study. And also I'd like to thank the people in the villages of Tutuila, American Samoa for allowing us to sample their reefs to conduct this study, and especially to Sunny and Ruth for providing us a home on Tutuila. I'd like to thank everyone in the entire Logan lab for being awesome, and the entire Moss Landing community for their support. And I would like to also say thank you to all of the different agencies that provided funding either to me or to the project. And just to go into it a little bit more, thank you so much to these three great advisors. Without you guys, it would not have been possible to do this project. Thanks to Cheryl for guiding me throughout this project and throughout my time in the master's program. Her mentorship has really transformed me as a scientist, and I have learned and grown so much through this project, and that's really thanks to her. Thanks to Amanda, whose passion for invertebrates always inspires me, and whose ecology and statistics expertise was so helpful in this project. And finally, thank you to Max, who provided key insight into pollution and water quality that was really important to this project as well. And then a huge shout out to these two right here for helping me do all of this field work in American Samoa. Jennifer Grossman and Casey Juliuson, they were so instrumental to this field component. We did all of that work in just 3.5 weeks on Tutuila, and that involved a lot. So thank you so much to both of them. They spent long hours with me searching for corals in the dirtiest water possible, staying up late, staring at corals, doing nothing in this heat stress assay, lugging around so many suitcases, and doing all of this while surviving on grilled cheese and fruit snacks. So thank you all so much for helping out. Another big shout out to the amazing lab mates and all the friends that I've made at Moss Landing and at CSUMB. The Logan lab is made up of some really inspiring people, and I could not have done this project without your support. An extra shout out to this group up here for spending a lot of time grueling away at the cDNA library preparation. You guys made it a lot more fun. And then finally, thanks to all the other people in my life that have supported me over the past 3 years. There's no way I could fit you all into this slide, but know that I'm thankful for all of you. To my best friends, many of whom are back in Maryland or in other parts of California, but whose friendships are still just as strong, even with miles between us. To my partner, who's always there to support me, especially when times get stressful. And to my family, my dad and my grandpa, who are always excited to hear about how I'm doing and always there to support me. To my other grandpa, who was the reason I really started pursuing a career in research. and to my mom, who would have been so proud to watch me defending my thesis today. So thank you to you all. I could not have done it without you. And with that, I look forward to taking any questions you have, and thank you again for listening today.
All right, thank you so much for that fabulous talk, Melissa. It's now time for the audience to ask questions. And then, as I said, we'll do like one big round of applause where we'll unmute and turn our videos on after the questions. So at this time, you can go ahead and use the raise hand feature if you have any questions for Melissa about her work. All right, Juliana, go ahead.
So first of all, I just want to say absolutely amazing job, Melissa. You did so much work on that and presented it very well. But my question for you was, I noticed that at your 2 low pollution sites, you found some pretty big differences in thermotolerance. And I was wondering if you had any ideas on why that might be.
Yeah, great question. Yes, I didn't mention this in the presentation, but I did mention how we didn't see differences in thermotolerance by pollution level. But yeah, if you remember, there were those 2 differences between the 2 low pollution sites. And it's hard to know exactly what is causing those differences, but one idea is that one site, Vitia, was on the north side of the island, while the other site, Fangatele, was at the very south side of the island. And past research has shown that these reef types are like pretty different, and there's a lot of other environmental differences at these 2 different sites. So it's possible that that might be underlying why we're seeing differences in thermotolerance, though it's hard to say for sure.
Great. And then I see GJ's iPhone. So whoever you are, please go ahead.
Oh, sorry, it's Jason. I'm
hi, Jason.
on my phone this morning. Great presentation again.
Jason, I think we lost you there. If you could repeat the question, that'd be great.
Yeah, since you have the data in hand, I'm wondering if you had a chance to take a look at when you're doing your WGCNA analysis, whether you can see correlations with the Cladoidium, Durastinium abundance versus any of the expression cassettes that you've been looking at, i.e., are you finding markers that associate with the diversity of symbionts in your control conditions?
That is a great question. And so it sounds like you're asking about, does symbiont community correlate with gene expression patterns in our WGCNA? And yes, we did look at that. I didn't have time to present it today. There's a lot of things that we looked at, but we found that at control conditions, symbiont community did not correlate with gene expression patterns. But interestingly, during heat stress, symbiont community did correlate. And so there were certain genes that differed in their expression at heat stress between corals that hosted just Durastinium or corals that hosted a combination of Cladocopium and Durastinium. And I have to admit, I don't remember exactly what those genes are off the top of my head, but it is written in my thesis.
Great, and then I see a question from Nate. If you want to go ahead and unmute, Nate.
Hey, Melissa, great talk. Really enjoyed it, and congratulations on all your hard work. I wanted to just ask a couple quick questions. One, for the WCGNA modules, I was wondering, have you looked at all to see like how those modules themselves, like if there's anything specific you've noticed about the nature of them, like are the ones, I think that something that might be interesting to do is to look at the genes that are in there and see if the kinds of networks that they're identifying have any similarities or differences? Like, are there lots of members of certain modules that are significant? Are there certain genes that have lots of connections to other things? Like, do you find hub genes, like genes that are controllers, in significant ones versus non-significant ones? Then I was also just a little bit curious if you could tell me, like, is the nature, think about multiple stressors, when we think about those pollution sites, is the nature of the types of pollution that they're experiencing similar or different? Is it like more like oily, like diesel fuel and things like that near the airport, and more industrial waste at the cannery, like, and how you think that might intersect with this idea that multiple stressors affect corals in possibly different ways? Yeah.
Two great questions. Okay, so for the first one, for the WGCNA modules, yes, it's something that I didn't really touch on too much in this talk, but like those modules aren't necessarily the same size and they aren't necessarily built up like the same way. So I think it is possible to look at the connections within those modules and see like which genes are kind of like driving those differences. And that isn't something I've done yet, but it is something that I can do in the future. I think I have some ideas on how I might get started on that. And then secondly, your question about pollution and how like pollution might differ at these sites. Like I'm just calling them high, medium, or low pollution, but like that might not be capturing all the variation that we're seeing, which is a great point. And it is really important too, because some of the discrepancies that I showed in those studies where like, pollution might increase thermotolerance or pollution might decrease thermotolerance, that can be explained, at least in part, like past studies have shown that like if there's more nitrate versus nitrite, or if the phosphorus ratio is different, like that can affect thermotolerance. So what would really strengthen this study is if we had better data on like the exact nutrient concentrations at these sites and like how they differed, as well as like other environmental variables that might differ at these sites as well. But unfortunately, we don't have a ton of that data, but future studies could consider that more.
Thanks. Great job.
Thank you.
All right, and then I see a question from Holly. Go ahead, Holly.
Hey, Melissa, you did a great job. That was a really awesome presentation. I think I just wanted to kind of piggyback off of what you already just answered a little bit with Nate's last question. And I just was hoping maybe you could just elaborate a little bit more. Where do you think the best next directions would be for these type of studies? Like taking these data that you've collected, what do you think would be the best next application, the best next questions to ask?
Yeah, that is a great question. I've definitely been thinking about that already too. Yeah, I think the point that I just made answering Nate's question is definitely a part of it. Like, I think that we could have redone this, or someone else could redo this with more sample sites and more environmental data to kind of see if we can capture what is driving these differences in baseline gene expression. Because I think that's really kind of where my mind goes for the next question. Like, we see these differences in baseline gene expression and it doesn't really look like pollution is causing them, but maybe it's some other environmental variable, or maybe it's like some evolutionary variable, like maybe there's local adaptation or something like that. So I think it would be cool to involve genomics in the study, like looking at differences in the genomes of these corals as well.
Awesome.
Yeah.
All right. So I'm not seeing any other questions right now. Maybe I'll just give it another second to. All right. Well, as I said, Melissa is going to be starting a PhD program in Australia in a couple months, but because she can't actually physically be in Australia, she will be around here in Monterey through this fall semester. So I encourage you to chat with her more about her research and her findings over the next few months now that things are opening up again. So with that, I'd like to invite all of you to unmute and share your video so that we can give Melissa a huge round of applause. All right.
Yeah, great job.
Good job.
You did it.
Crushed it. Nice job, Melissa.
Thank you.
Good job,
Melissa, that was so good. Good job. I hope my thesis defense one day is as good as that. So good.
It will be.
Honestly, goals.
Thank you all.
Great job, Melissa.
Thank you.
Yeah, great
That
job.
was amazing. That was amazing, Melissa. I just wanted to tell you all the way from Maryland. It was wonderful to watch your progression on this. So proud of you.
Thank you. Thank you so much, Gina.
Just wanted to say that you did a great job at explaining all that gene expression stuff to folks like me that don't work with this on a daily basis. So that really clarified a bunch of things. So really good job, Melissa.
Good. I'm glad to hear that. Thank you.
True. Your science communication game